Okay, so hello everybody, welcome to a biology lesson. We haven't done this topic for a very, very long time, but well, we are back to it. We were doing physics a few days later. As I said last time, we will be doing lots of things, very exciting things in biology, but we have to get like all the basic boring stuff first, and then we can move on, which is like very important, you see. Today we are back to DNA, as you can see from our topic. So what is DNA? DNA is actually the genetic material in your body that gives your body instructions on how to reproduce and on how to reproduce. It does that let me type this. DNA is genetic material in your cell's nucleus, which contain info on how to reproduce. There are actually DNA in viruses too, which, as I said, probably in our first lesson, are not really living organisms. But we'll talk about that later. Firstly, we need to know the structure of DNA. That's our first topic, yes. You might say, oh, it's double helix. And thank you very much, you are correct. It's actually made up of two strands. Let me get my pen out. Let me draw this. So, sorry, the colors. This is the first strand. There's a gap here. This goes here, back here. Oops. Back here. Here. This goes back here. So that's the DNA. And you can see, like these parts, this overlaps this part. Something like that. So, hope you understand. And then I'll change the color. There are bases like this. And they should be in different colors, but I don't bother, really. I not bother drawing them in different colors. It's just time consuming, like this. And probably in the KS2 or KS3 syllabus, that's all you learned about DNA. But is that really everything about it? No. The structure of it? No. So let's dive in a 2D model. The 2D model is actually, DNA is a polymer. Is a polymer, which means that it's made of loads of monomers, which is nucleotides, which we are going to look today. Firstly, there's a phosphate in the nucleotide, which is linked to a sugar. And this is the sugar. Let me draw this. And then the sugar is linked to a base. But there are loads and loads of that in DNA. So what happens is the phosphate is then bound, another phosphate is then bound with the sugar, which is then bound to another sugar, which is bound to a base. So it's like this, really. And that's one side of DNA. And the other side has a base, just like this one, and they're linked. And then it's completely the same as this stuff, although it's symmetrical. So, let me still gonna change the color. If this part was this big 2D model here, so the phosphate and the sugar is this part, and the bases are these parts. Okay, let me clear my drawings. The thing is that how do bases link? So there are actually 4 types of bases: A bases, T bases, C bases, and G bases. So A bases always link to T bases, T bases link to A bases, C bases always link to G bases, and vice versa. So this is very important. There are only 4 bases, and since all the nucleotides are the same in every other way except this, even the phosphates and the sugars are the same. There are only 4 types of nucleotide: A nucleotide, T, C, G nucleotide. And that's very important because this gives the DNA, and I'll move to the next page, back to writing again, its own genetic code. So every species, types of species, and thing and animals or like things in every type of species have different genetic code. And that's important because like your DNA could be a little different from your mother's and so on. Well, I think it's safe to say that they're mostly the same. And of course your genetic information will be way different from one bacteria. For a bacteria. So let's go on to our next topic, which is the making of protein. So how do you make protein? You know that cells, they make protein, they create protein by themselves, yes. But how exactly is that process done? So firstly, cells read the triplets in the genetic code. So triplets is every 3 things in the genetic code. For example, a C base, a D base, a T base code for CAT, which then would code for a specific amino acid. And as we know, amino acids linked together will become a protein. That's some basic stuff. And also, well, every genetic code, every 3, they can make a specific protein, which leads us to our second topic. The cell combines the amino acids and what happens is the DNA codes for the amino acids, which finally the amino acids combine to make protein. So, taking the fact that there are loads and loads of amino acids out there, and each of them have a different code, so the proteins made can be really different. The amino acids kind of like scramble up. For example, if you had a triangle, a circle, a rectangle, and a hexagon, there are loads of ways to put them together. Not to say that there are loads of each shape and different shapes. Now let's finally go to the last part. Viruses. Oops. Sorry, I made a mistake. So, in COVID-19, during the pandemic, it's over now, you don't need to wear masks now, which is good. Anyway, during the pandemic, we had to do things like RNA tests, probably mostly in Asia, and we also had to do some of them in Europe countries. What they do is they take some type of like cotton bud, and they put them in your throat, and then they take it out, put them in a glass tube or testicle, and boom, you get your result, positive or negative, depending on whether you have the virus in your body. So how does it do that? It's kind of like magic to most of us, but now I understand the reason. Once we've learned about DNA, and you probably need to look at the reason too. It's actually amazing. So firstly, we need to know is that viruses, although they are not living, some syllables say they are living, but they are not. They do not have cells, they cannot move, they cannot reproduce by themselves, so they are not living. They are actually pseudo-living, which means they are kind of fake, not living, which means they're still not living. Haha. So, it's not living, but they have DNA. Which, for example, if you take the DNA of a virola, which is kind of like smallpox, it actually has the code of entering a host cell's body, infected, make samples of itself and copies of itself, go to the thing's brain, and affect it even more. And these are one of the most complicated viruses, the variola. But there are other types as well. Some viruses are very simple, others have more complicated instructions. A strand of DNA is called chromosomes, so viruses have many chromosomes. So, we know that viruses cannot reproduce on their own, so they have to hijack host cells to make copies of themselves by letting the host cell create amino acids, proteins, where we have just seen it before, using RNA, not DNA, because the DNA cannot be taken out of the cell. Anyway, we'll talk about that maybe next class. It's very important. So, they create copies of themselves. And then this cell would be then stored straight there until too many copies are made and the cell self-destructs, or the virus goes out and infects other cells. This is the process. So, what we do in the RNA test is, firstly, we take out samples of our cells. And then, when you put it in the testicle, it actually tests or reads the genetic code. It does that by doing something by like putting things in and checking whether it's the code. I don't really know about this, and you do not need to know. All you need to know is that it can read the genetic code. And thirdly, what happens is that if the genetic code equals the virus's code, it is positive. Else, it is negative. So that's how these RNA tests can save our lives. It saves millions of lives by helping people know whether they are positive or negative, and then put them to specific places to stop other people from getting infected, which is actually theoretically a very good idea. That is everything you probably need to know about DNA. I hope you have enjoyed this video, and we'll see you next time.